start recording. All right, so now I'm recording. All right, so we need to get back into carbon fixation and some of the unique strategies. I just want to, full disclosure, I'm a little distracted. I should not have checked my text in between classes, and I found out that a family member, my sister-in-law's mom, has passed away. So I'm a little off balance right now. So please, I apologize if I'm a little off my cadence here, and hopefully I won't make any mistakes. If someone catches me saying something incorrectly, please pipe in and correct me. All right, so I'm going to do the best I can here, but it was a kind of a shock. So anyways, we are looking at carbon fixation, and I think we left off. I'm gonna, whoops, share my screen. And do that. Yeah, I'm gonna do it the other way. Sorry. Let me do it the other way. All right. So let me adjust. So our you just one big screen, one big slide. Is that what you're seeing on your end? Okay, thank you. All right. So we were looking at the C4 cycle last time. So I just wanted to put this up. This is something that you saw already, but I just wanted to recap this. And the idea now that these C4 plants have the ability to like tack on this extra cycle, the C4 cycle. So all plants do C3, but then these C4 plants, they evolve this extra cycle that they link into the C3 cycle. And it's a way, basically, when you look at it, it's a way for these plants to make sure that the Rubisco enzyme is not seeing too much oxygen under their natural habitats that they have evolved to live in. And so when you tend to find these plants in hot, consistently hot, year-round, hot, sunny conditions, droughts can happen. It's not always lush rainfall. And so they have this ability to shut down their stomata pretty nicely so they can, and which of course limits how much CO2 is coming in. But because they're so efficient at making sure that CO2 is delivered right to Rubisco, and Rubisco is like bathed in CO2, if you want to think of it that way, that it's just continually seeing lots of CO2 around Rubisco. So because they're so efficient at this, they can shut down their stomata when things are, hot, dry conditions, and they can still do a good job with the photosynthetic process and making their sugars. So here you see CO2 coming in, and instead of reacting with RUBP at first, what it does is react with phosphoenolpyruvate. So PEP is the acceptor molecule in this case when you look at the C4 cycle. And then it's the PEP carboxylase, or the pepcase enzyme, which is very picky. It only wants to react PEP and CO2 together. It doesn't care about oxygen. And then you get oxaloacetate, which gets converted to malate, which then malate I like to think of as a vector. It is a way to get CO2 over to where it's needed. It's just that it's tacked into this molecule that is malate. Malate comes over into the bundle sheath cell. The CO2 gets cleaved off during the decarboxylation event or process, and that's where CO2 is going to get fed into the Calvin cycle. So you see that the bundle sheath, most of the Calvin cycle is taking place in the bundle sheath. There's just that one phase that gets shifted to the mesophyll cells instead. And so really the bundle sheath is kind of devoted to, let's run the Calvin cycle. And then the mesophyll cells are like, well, let's do the light reactions. And that works out well for these plants because it allowed them to better adapt to their particular native habitats. After that decarboxylation, then you have to regenerate more of the acceptor molecule, that PEP. So here you see pyruvate shifting back into the mesophyll cell, where you have to expend energy to push this reaction and end up with PEP. So definitely there is an energy cost, extra energy cost for the microbes to do this. And where is it? Oh, here. The energy cost. Whoops. So you have 2 extra ATP for every CO2 that's coming in to run the C4. So then if you look at the total C4, C3 cycle combined for these plants, it's 5 ATP and 2 NADPH. And so, yes, there's a cost, but apparently the benefits outweigh the cost because many plants have adopted this strategy. And then we did talk about the idea that they're pretty good at parsing out where certain parts of the Calvin cycle take place. And so let me pull that up. So I think we left off right around here where we're looking at the idea that the initial part of the Calvin cycle happens in the bundle sheath. So when CO2 comes in and reacts with RUBP, with Rubisco, that does happen in the bundle sheath and you get your PGA in the bundle sheath. But then the PGA shifts to the mesophyll where you get the reduction phase of the Calvin cycle because that's where you have to use ATP, you have to use NADPH, and all that's coming from the light reactions. And so the mesophyll cell is cooking along, doing the light reactions, getting lots of ATP, NADPH, but in that whole process, they're also producing a lot of oxygen. And Rubisco would be at risk then, right? So keeping Rubisco in the bundle sheath cell, pushing the PGA out to the mesophyll cell where there's plenty of ATP and NADPH, let the reduction step happen in the mesophyll, then take your G3P back into the bundle sheath and finish off the Calvin cycle. And that is another kind of fine-tuned strategy that these C4 plants do in order to really make sure that you can get the Calvin cycle to go to completion and Rubisco does not see too much oxygen around it. So it's really an exquisite little strategies that they've evolved in order to better adapt to their particular habitat. So when you put a C4 plant in a tropical or subtropical habitat where they're native, that's what they evolved to live in, and then you put a C3 plant, side by side, head-to-head competition, in a tropical environment, the C4 plants typically win because they're just so much better at handling hot, dry conditions. C3 plants tend to do way too much of the PCO cycle, which is, again, experimentally, if you push a plant to constantly or very frequently do the PCO cycle, you see that they don't grow so well. Their growth rate slows down. Their overall growth yield upon harvest or at their mature stage is not as good. And so C4 plants win, so to speak, in a head-to-head competition if you have them in their native habitats. So what kinds of plants are C4 plants? There are monocots and dicots, so it's not limited to one or the other. So the monocots and dicots, remember, these are the angiosperms. Lots of different grasses. The Gramineae family has many species. Sedges family, Cyperaceae is sedges family. Chenopodaceae has been studied, that family. So I'm listing off some families. You don't have to know those by heart. What the big point is that you have a variety of different plants that can do that. It's not just limited to one family. They estimate about 1% of all known species are capable of doing this. And when you look at plants that are important to us in terms of food production, corn is a huge one. so maize or corn. Sugarcane, of course, is a huge commodity. Sorghum is showing up a little bit more in human food, but it's also used to feed animals in the animal husbandry, raising, animals for food production. Millet, again, that is more for feeding other animals, but sometimes I see it in some food products. Unfortunately, this next bullet point is pointing out some of the ones we really don't want to do good photosynthesis. Crabgrass, Russian thistle, pigweed, these are all considered weeds, so to speak, and can be aggressive colonizers of habitats, thistle in particular, if you're not careful. I've seen thistle go wild. So these also do C4. And so if you have them in your lawn, crabgrass commonly shows up in your lawn, and it can really start to outcompete the typical grasses we have up here, because most of the grasses we have here are C3 plants. And so crabgrasses can kind of, sometimes they can, under the right summer conditions, they can get a little out of hand in the right areas of the geographic areas. Okay, so C4 plants certainly are have some really nice, unique strategies. The other pathway I want to mention is going to be called the CAM cycle, or the CAM pathway, I guess. And luckily, it's very similar to what we just talked about. And so a lot of the same players. But when you're studying this information, of course, I really want you to know the C3 cycle very well. The C4 and CAM cycles that we'll talk about, you'll see that there's a lot of similarities. And so it'll be easy, I think, to tack these ideas into your brain. And a good way to study these will be for the C4 and the CAM, will be if you can kind of study them side by side and ask yourself, what's similar and what's different between the C4 and the CAM, because that's probably how I'll ask you to think about it on the exam. Show me what are similar parts, show me what's different. All right, so let's get into this other cycle, the CAM. And so that stands for crassulacean acid metabolism, which thankfully abbreviates down to CAM. So this was discovered in the family Crassulaceae, and CAM plants are really nicely native and well adapted to desert environments. So think about, Arizona and New Mexico, Nevada area, these types of environments where the temperatures can soar well over 100 routinely. Drought can be severe, although they do have the monsoons sweep through if everything's going okay. So it's not like they're totally without rain, but it's very much, you'll have your downpours of a monsoon, and then you'll have months of dry. And so it's not the same thing as tropical and subtropical. So the family Crassulaceae are a lot of succulent plants. Think about like aloe vera and the giant saguaro cactuses or cacti. I don't know what the plural is of cactus. Cacti. And so you have these plants. And so when they first discovered it, it was in the family Crassulaceae, and a little bit unfortunate because we now know it's not just this family of plants that do this. So unfortunately, by the time they figured that out, they already had this jargon established, so they didn't stop it. And so we still refer to them as CAM plants. But anyways, so again, just like with C4, they have a C3 cycle as well. So think of the CAM pathway as an addition and kind of linked into the C3 cycle. And when you look at how plants run the CAM cycle, basically it does a couple of things for them. It really allows them to close down their stomata appropriately so that they can conserve water. So they are really good at conserving water. And this cycle or pathway also allows them to make sure that Rubisco, the Rubisco enzyme, is seeing CO2, CO2, CO2. So it concentrates CO2 around Rubisco so that oxygen can't get an upper hand, so to speak, in the competition for the active site of Rubisco. So we say that the CAM pathway has a temporal segregation instead of a physical. Like the C4, that was a physical segregation of where these reactions are happening via the, both the mesophyll cell and the bundle sheath cell. With the CAM plants, now we say it's temporal, so it's time. There's what happens during the day. There's certain things that happen during the day, and then some other things happen at night. So we need to think about that. So at night, if you think about it, if you're a giant saguaro cactus living in the middle of nowhere in Arizona desert, and it's hot and sunny, hot and sunny, hot and sunny every single day, not a cloud in the sky every single day, it doesn't make a lot of sense to open your stomata during the day because you'll dehydrate so quickly and die. And so they have adapted to these hot desert environments by closing their stomata during the day and doing some things, important things during the day, even with closed stomata. But then at night, open up. So at night in the desert, if you've spent any time there at night, if you've been lucky enough to experience that, the temperatures drop. The humidity tends to go up a little, anyways, a little bit. And of course, with the temperatures dropping, when you open your stomata, you won't transpire as much water out of your body. And so it makes a lot of sense to open your stomata at night. That's when you should take in your CO2. And then because the Calvin cycle needs sunlight, you can't immediately start running the Calvin cycle. So there's a conundrum. And so the adaptation for them, for these plants was, okay, I'm going to open my stomata at night. I'll take in all that lovely CO2. I can't run the C3 cycle right away because some of those enzymes need light to be fully activated. So I'm going to store the CO2 overnight. And the vacuole is an awesome place to store stuff in the plant cells. So they stuff it into their vacuole all night long and store it up. And then sunrise happens in the desert and it gets bloody hot and bright sunlight, no clouds. So then they close down their stomata and then they say, okay, now with the sunlight hitting my body, I can take the CO2 out of storage and shift it over to the chloroplast and get Rubisco up and running with the Calvin cycle. And so that's what you see. So let's work through this. So the night reactions, you have carbon dioxide coming in, stomata open up at night, CO2 comes in. When it dissolves in water, it forms the bicarbonate HCO3 minus. It reacts with PEP again, just like what we saw with the C4. So PEP is the acceptor molecule again. Again, you get oxaloacetic acid, OAA, which then converts to malate. So all of that should sound very familiar. The same enzyme, PEP carboxylase. So that's great. Got that same lovely enzyme that does not care about oxygen and will only accept in CO2 and PEP. And then if you are curious about the enzyme that does the next step, it's NAD malate dehydrogenase that forms the actual malate. I won't ask you about that enzyme. Pepcase though, make sure you understand pepcase is the one that's interacting with CO2 and the PEP or the PEP. Malate gets made. Okay, so again, it's going to be like a vector for CO2. You get that malate molecule, but again, the plant can't immediately do the C3, so they stuff all that malate into the vacuole. And malate is an acid. Malic acid is another way to say malate. And so here in a visual form is what we just described in words. So you have CO2 coming in, reacting with PEP, phosphoenolpyruvate, in the active site of the PEP carboxylase enzyme. You got oxaloacetate is the first product, which then converts to malate, and then they stuff malate into the vacuole. So that's all happening all night long. And if you stick a pH probe into the vacuole, which they've done, they can show that as malic acid keeps coming in all night long, the pH of the vacuole keeps going more and more acidic, which just means that there's plenty of malate getting stored up. So all of this is happening during the night when the plant has the ability to go ahead and open the stomata and take in enough CO2 without losing too much water through the process of transpiration. Okay, so now the trick will be when daylight comes, you have to get that malate shifted over to the chloroplast. So now the decarboxylation step, that just means you need to cleave off the CO2 from the malate. So you have to physically shift the malate, but it's all happening in the same cell, all right? This is all the same cell. You get malate coming out of the vacuole and getting over to the chloroplast. And then when it gets to the chloroplast, it'll get decarboxylated, and the CO2 can then immediately get used by Rubisco to react with RUBP and do the C3 cycle or the Calvin cycle. So that's great. And then the other idea is you get pyruvate as one of the other products again. So then in this case, there's a difference. The pyruvate will actually get stuffed into the chloroplast and it'll enter a pathway that forms starch. So it's not really a cycle. So sometimes I catch myself saying CAM cycle, but it's really more of a pathway because it's not a true cycle like what we see in the C4 plants. So let's take a look at this now visually. During the day, this is what's happening. So during the day, again, if you're a giant saguaro cactus, you want to shut down your stomata to conserve water. CO2 can't come in, but that's okay because you have all that lovely malate in your vacuole. So get the malate mobilized. Again, it's like a vector, moving the malate out of the vacuole, get it to the chloroplast, decarboxylate it. CO2 can get into the Calvin cycle. Rubisco's happy now, sees lots of CO2. And then the other product, when you cleave off CO2, is pyruvate again. And then you see here, they just show it gets used in a pathway to form starch. And then let me go back. And then you can see at night, you can see that the starch starts to be broken down to form the phosphoenolpyruvate. All right, so it's not truly a cycle. It's more of a pathway that we're talking about here. C4 cycle is a cycle. Questions on the CAM? No questions? Crystal clear?
Yep.
Okay. Okay, so one thing that's important for your own knowledge, and I take advantage of this knowledge quite a bit at home. Some CAM plants are what we call facultative, and it's important to be aware of this. So there are some species that if, for example, aloe vera, I have aloe vera at home a lot in case I burn myself when I'm cooking, which I tend to do a lot. And so I use aloe vera on my burns from the oven. So plants like this, if I have them at home, I have a lot of succulents actually in my bedroom for a particular reason, because they open their stomata at night, right? So as you're sleeping, if you're lucky enough to get anywhere beyond 5 hours of sleep, if you think about it, people hopefully are sleeping 8 to 10 hours a night, which would be lovely, but unrealistic for a lot of us. And so you're breathing out all this stuff all night long, including CO2, of course. And so if you have CAM plants in your bedroom, they're taking all that in. The problem is many CAM plants in pots in a person's home, they're facultative. And so if you give them plenty of water, they'll just say, you know what, I have plenty of water. I don't see any issue here of why should I run the CAM cycle when it's an extra, or CAM pathway, when it's an extra, effort for me and extra resources I have to expend. And I've got all this lovely water. Whoever's home I'm living in, they're so nice to me. I'm just going to shut down the CAM pathway and I'm just going to do C3. And so then it kind of defeats the purpose. If you have a bunch of succulents or CAM plants in your bedroom and you're too nice to them with your watering, they won't open their stomata at night. So please be aware of that. If you are deliberately trying to put plants in your bedroom to clean up your air quality, if you have succulents in there, or CAM plants, I should say, make sure that you are kind of mean to your plants. Don't water them as much as you think you should. Stress them a little bit so that they're forced into doing the CAM pathway. And so I have two, I think, in my bedroom right now of all the plants in there. And so I just water them maybe once a month, once every 3 weeks, depending on how you have to, kind of take into account the humidity in your room as well. How humid is it? So yeah, I'm kind of mean to them, but they're survive. They seem happy. And actually, most people overwater CAM plants in general, and they get root rot. They're not used to having their roots wet all the time, and they get root rot, especially if you have them in a pot. It's very easy to overwater, so be careful about that. So yes, Crasulaceae family does have many species that are CAM plants, but it's not just the Crasulaceae. So unfortunately, the name is a little confusing. And then if you look at a family of plants and you look at all the species that belong to that family, you'll see a mix. Some do CAM, some don't. And then you can see in one family everything, that some are doing CAM, some are doing C4, some are just doing C3. And so it is kind of all over the map. This bullet, the cacti, the I guess that is plural then, I hope. Euphorbias, pineapple, vanilla, agave, they all do CAM pathways. And of course, pineapple, obviously a huge food commodity. Vanilla is very, very popular. It's not really an herb. It's not really a spice. I guess it would fit more in just
vanilla is a spice. It comes from the seed pods from a plant.
Yeah, I guess it would, by definition, fall into the spice category. And it's actually from an orchid. It's the only orchid that is farmed for a food-related product. It's the only orchid. Agave, what does agave give us?
Tequila.
Yeah, lovely tequila. Yeah, that brings me back to grad school days. Lots of agave tasting. So billion-dollar industry, I'm sure. And I found out it's kind of like when I first found out that different beers taste differently. I used to think, oh, beer probably all just taste the same. Well, I found out tequila, there's definite differences in the taste of tequila. So if you're over 21, you can certainly sample a few and convince yourself. Anyways, so CAM is also found in more primitive plants. And so when you look at the evolutionary history of these extra pathways and cycles, they think that, the first to evolve was the C3, of course, because all plants do C3. So that was first on the scene, and actually microbes invented that. And so the C3 cycle, and then the next to come along on the evolutionary timeline was actually CAM, and then finally the C4. Shots for science. Yes. I'd be game for that, especially today after getting that bad news. So not that I'm promoting alcohol, but in moderation. All right, so what else? We have, maybe a bonus question. I can devise something to do with a shot for science. Let me think on that. Anyways, what's the comparison? So they've done some nice measurements and found that CAM plants win in terms of their ability to conserve water. Look at that, 50 to 100 grams of water lost. So that means they're really good. They don't lose much water. C3, on the other hand, look at how much water they lose. So it's an issue. So again, C3, because of Rubisco being sloppy, C3 can, if Rubisco is seeing too much oxygen, or if the CO2 levels drop, which means then oxygen can get into the active site a little bit more, you're going to run the PCO, and that's not as efficient. And with water loss, these C4 and CAM plants, really what they're doing is they're shutting down their stomata under hot, dry conditions, so that you either shut it down all the way or greatly reduce the aperture of the stomata. So again, but they're okay because they are so efficient because of the C4 and CAM cycles. They're so efficient that they can still do photosynthesis appropriately. So in the order of evolution, they think, of course, C3 happened first, that cycle, and then along came the CAM pathway, and then finally the C4 pathway, or cycle, sorry, evolved and took hold with some of the angiosperms. All right, so let's see. A little bit about pepcase. It's a very nice enzyme because it doesn't care about oxygen. They've been studying it and looking at, what's controlling its activity. And they see that as malate levels go up, it tends to slow down pepcase activity. So that's one thing they've noticed. And then what happens is there's a phosphate group will get stuck onto one part of the enzyme where there's a serine amino acid, so onto the pepcase enzyme on the serine residue. And under those conditions, that's telling the enzyme get fully active and get going. All right, so again, this idea of a phosphate group as a signal to tell the molecule or the plant to do something. And so that's kind of a nice strategy that many organisms actually have. Yeah, I like the chat. All right, so here visual inactive day form. So there's no phosphate group here. So that's telling the enzyme just chill out, kind of take a nap. During the day, they don't want to open the stomata, so there's really nothing for pepcase to do, right? No CO2 coming in. So during the day, take a nap. And so that lack of a phosphate is pretty much saying just chill out. And then at night, you can see you have to expend some energy. All right, so there is a cost to doing the CAM. There is extra resources and energy involved. And you can see there's a phosphate group that gets tacked on, and that's waking pepcase up and saying, okay, time to get off the couch. Let's get cooking here and take CO2 in, react it with PEP so that we can start making a bunch of malate. Okay, what else? So that's the C4 and CAM. As you can see, there's same players that you need to think about. So that should make studying this a little bit more streamlined. And again, do that compare and contrast what's similar, what's different as you're studying, okay? So we have been talking about the C3 and the C4 and the CAM. So they're making, ultimately, if the Calvin cycle or the C3 cycle goes well, you end up with either sucrose or starch. If the plant makes the decision, I'm going to build up my starch storage, my starch supply, that all happens in the chloroplast. So, and that's where the C3 cycle is happening. So there's no movement. Everything just stays there and they start making chloroplast, or I'm sorry, starch. If they want, some plants, they don't really do much starch storage. Some instead pack sucrose into their vacuole for long-term storage, which is interesting. So they find that in some crop plants. So oats, wheat, and barley, they've noted if it has a lot of extra sucrose that it can store away, it packs it into the vacuole instead of making starch, which seems weird, and I have not found a good explanation for that. So I don't know, and then the plant may say, you know what, I need to get sucrose made because I need some sucrose here in the leaf tissue, but I also need to send it through the phloem and, from source to sink, because my roots might need some sucrose and, other parts of the body. So then certainly the leaves need some sucrose. Hopefully they can store some sucrose in the form of starch usually. and then they have to transport it throughout the plant through the phloem translocation process from source to sink. And then once it gets to the sink tissue might have to use it right away. They might have to use it to repair damage. They might have to use it to grow. They might have to store it too. That they might be in the phase of their life cycle. For example, if you're making a carrot taproot where you need to store it up. Okay, so again, just thinking about the idea of phloem translocation, source to sink, and then what's happening in the leaf, source tissue still needs a share of that sucrose as well, but then they have to get the rest of the plant fed. So in a visual form, this is what's happening as the plant is making the decision, should I make starch or should I make sucrose? And we're not, oh, that's my office phone, I'm sorry. Can I mute it? Yeah, it worked. All right, so I'm not going to get into these pathways. I will not ask you to know these to any significant extent. There are a few things I want you to know. First of all, if starch is going to get made, know that it's happening in the chloroplast. If sucrose is going to get made, that's happening out into the cytoplasm or the cytosol. It's an either or idea. So once you have that G3P made in the Calvin cycle, where that's the triose phosphate here, once you have the G3P made, then, once you have the G3P made, it's either or. Either some of that G3P stays in the chloroplast and the starch pathway kicks in, or you get it transported out. And you see that there's this, they're pointing to this translocator. It's just a transmembrane protein that allows for the transfer of DHAP, which is just G3P rearranged into DHAP to come out, or it'll stay in. So let's look at this a little bit. Whoops. Okay, so if starch is the decision, the plant's like, wow, I got a lot of, I'm doing photosynthesis, I'm so efficient, this is great, I'm making tons of sucrose. In fact, I'm making so much, I can store some up as starch. That all stays in the chloroplast, and I just put arrow, starch. The other thing I want you to know is that it's going to require some energy to get this to happen. So there is an energy expenditure. If you're curious, one of the key enzymes toward the end is ADP glucose phosphorylase. And here, let me show it. ADP glucose phosphorylase is right here toward the end and as you're getting into the last stages. So I just wanted to show you the key step where you have to expend some ATP. You don't need to know the name of the enzyme, but know that starch synthesis is in the chloroplast, so that G3P will be pushed into this cycle or pathway. ATP has to be expended, but then you get lovely starch that starts to pack into the chloroplast. Great. Now, if the plant says, you know what, oh, here, lovely starch grains showing up in the chloroplast. So this plant was was cooking along with photosynthesis and had lots of extra sugar and could devote some of that sugar to starch synthesis. Now, sometimes the plant will say, you know what, I need to make sucrose. So remember, sucrose is two molecules, glucose and fructose, bound together. This also requires energy, and it's it's not ATP, it's UTP, which is weird. And it involves that transporter protein that we saw in the membrane of the chloroplast. So they call it an antiport protein because antiport means as something moves through in one direction through that protein, something else moves in the opposite direction through that same protein. So you get this opposing directions of movement of these molecules. So let's think about that a little bit. The fancy name for this protein is called the orthophosphate triose phosphate translocator. Good gravy. So I'm okay if you just type in antiport protein and know that it's in the chloroplast membrane. You don't have to know the full name. So what is this? What is it antiporting? What's going on there? It's exchanging a phosphate, an inorganic phosphate for a G3P, which is actually the DHAP form, but it's really the G3P. One of the G3Ps will get destined to make sucrose. It gets rearranged to DHAP. DHP comes out of the chloroplast into the cytoplasm, and then the inorganic phosphate goes out of the cytoplasm into the chloroplast. So we're zooming in right here to this translocator antiport protein. And so you're seeing the triose phosphate coming out as the inorganic phosphate comes in. So that's what we're zooming in on here. And then here in the cytoplasm, again, you don't need to know any of the players in here other than please know that UTP is the energy source that is used to push some of these last reactions. And you get sucrose finally showing up at the end, all happening in the cytoplasm. Okay, so this is, again, it's an either or process, and this antiport protein is exchanging that inorganic phosphate for a triose phosphate. So it is a competitive thing here, and either or, and it's competitive. So what do I mean by that with competitive? If there is low concentrations of inorganic phosphate in the cytoplasm, that means there's not a lot of inorganic phosphate available to do the exchange and move into the chloroplast. So that means a lot of DHAP is not going to be coming out into the cytoplasm. So if there's not a lot of inorganic phosphate in the cytoplasm, that actually means it's going to favor the synthesis of starch. The DHAP goes, oh, I have no way to get out of here. I need that antiport mechanism to kick in, but there's no inorganic phosphate out there. I'll just stay in the chloroplast and I'll be destined to make starch. And then of course the flip side, if you have lots of inorganic phosphate in the cytoplasm, then that can come through the translocator, which again means DHAP now can do the antiport and say, yep, I can come out into the cytoplasm and go ahead and make sucrose. All right, so it's this interesting little kind of competitive thing, and it really matters what the concentration of the inorganic phosphate is in the cytoplasm. And that's really all I'm going to say. They're looking at this very intensely. This has a lot to do with food production and trying to maximize yield and biomass. So there's been a lot of, and still is a lot of research going on in this, but it gets pretty complicated pretty quickly, and I don't have time to get into it. So big picture ideas when it comes to this idea, the idea here of starch versus sucrose, okay? Are there any questions on the starch versus sucrose or anything else?
I've noticed that some of these slides in your PowerPoint for this section aren't in the one that was on EDUCAT.
Oh,
Can you possibly update it?
yeah, I thought I did update. Yes, I will update.
Thank you.
Yeah. I think I'll update by taking this one out. We don't have time to get into this. I think I will, yeah, I will take this out. And then these last, there's several tables here. Again, this is just for study purposes. If this works for your brain, great, use it. If it doesn't work for your brain and it's not helping you, don't worry about it. I just put that in for study purposes. So these are, this is detailed of the Calvin cycle. And some people like to pick it apart like this, and that's great, that's fine. And this is the PCO cycle. And again, this one kind of goes on and on. C4 cycle, all right. And then starch, synthesis, starch, sucrose. All right, so this is purely if it works for you, if it helps you better understand something that you might be trying to grapple with, please use that. But if you don't find it useful, don't worry about it. Okay, the next topic is looking at signals from sunlight. We're going to start thinking about how the plant is perceiving sunlight and how does it behave when it gets that signal. But we're running out of time. I don't want to start a new topic right now. It's just literally 3 minutes left. So we'll call it good for today. And have a safe weekend. Be careful out there. But hopefully get out when it's not snowing or raining and enjoy some fresh air and the beautiful woods and the beautiful fall colors. All right. So have a good weekend, everybody.
Best wishes to you and your family.
Thank you.
Sorry for your loss. I hope you have a good weekend.
Thank you.
Our condolences.
Thank you very much. I appreciate that.
Do a few shots. Also, I have a question.
Yes.
The LHC